All right, welcome. I hope everybody is doing fantastic today. Let's talk about cells. Um, so I'm going to start with a little bit of background. I'm going to discuss some of the key differences between prokaryotic and eukaryotic cells. But the bulk of this chapter is going to be spent focused on eukaryotic cells and kind of going through basic structure and function of some of the different parts, the different organelles that are found in eukaryotic cells. And then at the very end, we will step back and just speculate a little bit about how eukaryotic cells may have evolved. Okay. And I'm going to have this divided into a few parts because chapter 5 is one of the longer ones. So let's start with some background talking about what's called cell theory. And so this theory, this idea that all living things are made out of cells, was really the first unifying theory of biology, meaning it tied together all different parts of biology. Um, now theory of evolution would be another unifying theory of biology. But cell theory really has three parts. So it says cells are the fundamental unit of life. You have to have at least one living cell to have a living organism. All organisms are made of at least one cell. And then the third part is all cells come from pre-existing cells. And there's a few implications of cell theory. One is it implies that the functions of all cells are going to be similar. So you may remember in chapter 1, I gave you kind of a short list of things that living organisms have to be able to do, certain characteristics they always have. And if all living organisms are made of at least one cell, then that implies that all cells have to be able to do everything on that list, right? So there are these critical functions every cell has to be able to carry out. So there has to be a fair amount of similarities between all different types of cells. Um, the fact that all cells come from pre-existing cells would argue that life is continuous then, right? Cells are not just magically appearing out of thin air. They come from existing organisms, existing cells. And then lastly, this would also imply that the beginning of life was the beginning of cells. All right, so as I'm sure you're aware, cells are small. Of course, I've got mostly in parentheses here because there's always a few exceptions to that. Like some eggs can be larger cells. But just to give you a feel for exactly how small, I wanted to go to this University of Utah Department of Genetics has this cool little cell size and scale illustration. Um, so we start with this image of some small but easily recognizable objects that you can see with the naked eye, and you can see we're on the millimeter scale here. So you've probably measured something in millimeters before. And then I'm going to grab this little slider down here at the bottom, and as I pull it to the right, we're going to start zooming in on this image. So we get a grain of salt. Um, and now here we are at a quote-unquote large single-celled organism. So this is an amoeba, large eukaryotic cell. And you can see our scale has switched to the micrometer scale. So there are 1,000 micrometers in 1 millimeter. So this would be about a half a millimeter in diameter. Another single-celled organism, paramecium in the background there. Then we have a large human cell, the egg cell. You can see the sperm next to it for comparison's sake. A skin cell, red blood cell. So some different human cell types here. We're down now at the low end of the micrometer scale. Here's another eukaryotic cell, Baker's yeast, Saccharomyces cerevisiae. There's a large chromosome, the X chromosome. And now we start getting into some smaller cells, bacteria, so E. coli bacterium, and then we're down to some individual organelles that we would find in some of the larger eukaryotic cells. Keep going here. Start getting into the virus realm here. Got the measles virus, excuse me, influenza virus, You can see our scale has changed yet again. We're now at the nanometer scale. So it'll be 1,000 nanometers in 1 micrometer, 1,000 micrometers in 1 millimeter. So small viruses, some smaller organelles here, the ribosome, um, hemoglobin, an antibody, etc. Then we start getting into some of the building blocks of some of those macromolecules we were talking about, right? Glucose, some amino acids, a nucleotide, and then we're really down at the atomic level here. And then you see the scale changed again with the picometer scale. So anyway, it's kind of fun to play around with this. You can always pull up the slider, but it gives you a sense for exactly how small cells are. So, hopefully you're walking away from this with the idea that, yeah, even the large cells, quote unquote big cells like the amoeba here, are still pretty freaking small. All right, let's get back into the slideshow here. Okay, so why are cells so small? We have some very large organisms on planet Earth, so it might seem like it would be a little bit simpler just to make them out of larger cells. Why do we need trillions of these tiny cells? And the reason biologists think all cells are small is because for them to function properly, they need to have what we would call a high surface area to volume ratio, meaning there would be a large surface area, basically a large amount of plasma membrane surrounding each cell relative to the volume inside the cell. And all the evidence we have suggests that this large surface area is important for cells to be able to exchange materials efficiently with their environment, send and receive signals efficiently as well. So it's all about being able to communicate and exchange material with their environments. And so just to give you an illustration what I'm talking about here, right, here's a large cell on the right. See the diameter is 200 microns versus a small cell on the left, 2 micron in diameter. We've got the surface areas calculated here for us. So, the, excuse me, the large cell does have a larger surface area, but then if we think of that in the context of the interior volume of the cell, the ratio of those two things is much larger, right? The amount of surface area relative to the interior volume is much larger on the smaller cell. So we think there is some upper limit in terms of how large cells could effectively be and still be able to function properly. Now, you may remember chapter 1, I introduced this idea of microscopes being critical to our understanding of cells and really a lot of modern biology. So I just wanted to go over a couple of key concepts related to microscopy. For any microscope, there's really two key measures that are important. One is magnification, how much is it increasing the size of the cell or the object that you're looking at. And then the second is what's the resolution? How clear is that image? And I want you to be aware of these two main types of microscopy. So there's light microscopy, where you're simply shining light through your sample, right? You're typically using glass lenses. This would have been, the very first microscopes would have been light microscopes, and they're still used today. And they have a pretty decent resolution, so you can get down in the 0.2 micron range. So that's pretty good. But now we also have what are called electron microscopes, where instead of shining light through your sample, you're basically hitting your sample with a beam of electrons. And these have a significantly higher resolution. So instead of 0.2 microns, you're at 0.2 nanometers. Remember, there's 1,000 nanometers per micron, so basically 1,000 times better resolution. So there's different types of light microscopy. The simplest form, which is what we typically do in Principles of Biology 1 lab, or even in maybe a microbiology lab, would be just bright field microscopy. So you slap your your sample that you want to look at on a slide, shine the light that's part of the microscope through your sample, and just look and see. You can see your cells there. Over the years, biologists, microscopists have developed different ways to try to make the image a little bit better. So there's certain techniques that will basically enhance the contrast in different ways. I'm not so worried about you knowing all these uh, different subtleties here. There's, of course, staining, right? Here it looks like we've stained the nucleus with some particular dye, so that makes that part of the image stand out a lot more. And now we have what's called fluorescence microscopy, where you can actually add little protein tags to molecules in the cell. And those proteins are fluorescent, meaning they will literally glow specific colors if you shine the right wavelength of light on them. So you can get these, really incredible, brightly colored images where you get great contrast and can see all the different parts of the cell very clearly. In contrast to that, excuse me, electron microscopy, as I said, has much better resolution, but it's always a little bit more boring to look at because you're always going to get black and white images, and you're always having to look at dead cells in electron microscopy. You have to fix the samples and oftentimes section them. So even though you have this very high resolution, in the back of your mind, you're always thinking, well, is this really what it would look like if the cell was still alive? Whereas in light microscopy, you can look at living cells. So really, these two things complement each other very well, right? We can look at the live cells by light microscopy. We can look at then dead fixed cells by electron microscopy, but at much higher resolution. And there's a couple of key types of electron microscopy. There's what's called a transmission electron microscopy, or TEM, it's sometimes abbreviated as. In this case, you're really looking at a cross-section through the middle of the cell, so you can see the interior contents, organelles, things like that. You could also do what's called freeze fracture microscopy, where you literally deep freeze the cell and then kind of crack it in half. So again, it's letting you see inside the cell. And then you have what's called scanning electron microscopy, where you're basically scanning over the surface of the cell, so you're getting just an image of the exterior. Okay, so we use microscopes to learn a lot of what we know about cells. Basically for 100 years, everything we learned about cells came from just observing them under microscopes and applying different kinds of stains, etc. So before we get into some of the different parts of cells, I want to talk just briefly about the difference between prokaryotic and eukaryotic cells. So you might remember 2 of the 3 domains of life were prokaryotic. So for sure there is more prokaryotic life on Earth than eukaryotic. Without a doubt, the first cells on planet Earth were probably prokaryotic. We have some fossilized prokaryotic cells that are like 3.5 billion years old. And you've probably seen this before, but one of the big distinctions between prokaryotic and eukaryotic cells is that prokaryotic cells are smaller, eukaryotic cells are larger, as we saw in that little slider illustration we were going through. But another big difference is that prokaryotic cells lack membrane-bound organelles. So when we talk about eukaryotic cells, we're going to look at all these different organelles, and I'm sure you've heard of some of these, mitochondria and endoplasmic reticulum and Golgi apparatus. All of these organelles are made out of membrane, and they are lacking in prokaryotic cells. There's no nucleus either in prokaryotic cells. So in that sense, prokaryotic cells, you could argue, are a bit simpler, but that being said, they still have to be able to accomplish all the same things that eukaryotic cells do. They just really don't have the advantage of these organelles to help them get it done. Okay, so as I mentioned, prokaryotic cells are smaller. We'll take a look at them here in just a second. A lot of prokaryotic life exists as single cells, but not always. Sometimes they can be clustered or in chains or groups, or something like a biofilm maybe would be a layer of prokaryotic bacteria. Um, but for sure, prokaryotes, as I just alluded to, are very, very biologically successful. So they way outnumber eukaryotic cells. You find them on all parts of planet Earth, even places where there's very extreme environments, high temperatures, weird pHs. I saw this stat. I can't remember where I found it, but it's pretty crazy. Which is that if you were to walk outside, scoop up a handful of dirt, they estimate there would be more prokaryotic cells, more bacteria in that one handful of dirt than the number of humans that ever lived on planet Earth. And that's insane to think about, but also think about how many handfuls of dirt there are on planet Earth, right? So we are way, way, way outnumbered by the prokaryotes. All right, so let's look at basic structure of a prokaryotic cell here. And as I said, it's pretty simple, so this isn't going to take us too long. But every prokaryotic cell is surrounded by a plasma membrane. A lot of them will also have a cell wall or sometimes a sugary capsule as well. So we get a cell wall layer, maybe a capsule layer. But there is going to be that cell membrane. So every cell is surrounded by a membrane, prokaryotic or eukaryotic. There's going to be cytoplasm inside the cell. There's going to be DNA, genetic material. It's just not going to be inside of a nucleus. I mentioned that prokaryotic cells don't have membrane-bound organelles, but they do have organelles like ribosomes, which are not membrane-bound. And ribosomes are the organelles that produce proteins. So, even prokaryotic cells have to have proteins, so they need those ribosomes. Even though there's no nucleus, sometimes there is a part of a prokaryotic cell called the nucleoid, which is kind of the region of the cytoplasm where the DNA might be kind of tethered or anchored and spend most of its time. And then we'll talk about the fact that some bacteria can move, they're motile, so they may have cilia or flagella to help them with that process. So for example, here you can see a nice image of a bacterium taken with an electron microscope. This would be scanning EM because we're looking at the surface of the bacteria. You can see this one, I guess it's got places to go. It's got a bunch of flagella here, and you can see these little, these are actually pili rather than cilia, little extensions on the surface of the bacteria that can be important for attachment, or there are even specialized pili that will allow bacteria to kind of exchange genetic material with each other. But we will come back and talk about prokaryotes and bacteria at the very end of the semester, so we're not going to dig too deep right now, but I did want you to be aware of those basic structures that you find in a prokaryotic cell. So let's talk about eukaryotic cells now. And as I mentioned, they're going to be larger than prokaryotic cells, up to 10 times bigger. They do have those membrane-bound organelles, things like the nucleus and the mitochondria. And if you're not familiar with this idea of organelles, you've never really thought about, well, why do they have these organelles? What's the advantage of having those? Really you can think of each organelle as a little mini organ, right? We all know our body is filled with organs. Each organ has a specific job, right, that helps our body function properly. And it's really the same idea with these organelles. Each one is a little compartmentalized mini organ that has a very specific function and helps that cell do what it needs to do more efficiently. So prokaryotic cells can still do the same things eukaryotic cells do. They're just generally not going to be as efficient because they don't have these organelles. So before we dive deeper, I just wanted to do a quick comparison while we're talking about this sort of introduction to eukaryotic cells. Quick comparison between an animal cell and a plant cell, and talk about what you might see that's different between the two. But mostly here we're going to focus on different parts of plant and animal cells, so the different organelles. So we got our cartoon of the animal cell here. You can see all the different organelles labeled. And there's really two that I wanted to focus on here. Well, actually, really just one that I want to focus on in animal cells that you do not find in plant cells. And that is these little guys here called the centrioles. And these are not organelles that get talked about as much as some of the others. But centrioles are basically important for growing microtubules that are part of the spindle during cell division. So if you've ever studied mitosis before, and if not, that's okay. We're going to look at it in an upcoming chapter. You have this sort of web of filaments that's called the mitotic spindle that pushes and pulls the chromosomes around. And that web, that network, grows from these little organelles called the centrioles. So it's important for cell division, basically. And interestingly, plants, as I just mentioned, don't have centrioles, but they do still carry out mitosis, and they can still form a spindle. They just do it in a different way. And so then you can see here's our cartoon of the plant cell. It obviously looks a little bit different, despite the fact that it does have a lot of the same organelles. But I wanted to focus on differences here. So one first thing you might notice is the plant cell has this structure called the cell wall outside the plasma membrane that animal cells do not have. Right? It's a dense, tough, protective layer, right, made of cellulose, one of those key carbohydrates that we talked about in previous chapter. Of course, there's chloroplasts, right? Photosynthetic organelles. Animal cells do not have chloroplasts. There's also vacuoles. Most animal cells are lacking vacuoles. There's a large fluid-filled cavity inside the plant cell. Also peroxisomes, fairly unique to plant cells as well. All right, so I'm going to stop there, and we pick up in part 2. We will start going through some of these different organelles, just basic structure and function.